So, good morning, good afternoon, and good evening to all of you around the world. Today, I'm going to talk about the story of Tom and Jerry. Don't be surprised. This is how pathogens work inside our body, or more precisely within our cells. We often end up with having multiple pathogens within our cells, like Tom and Jerry living within the same house. They either attack each other and or live harmoniously with each other, even if it is temporary. Under both conditions, our cells are damaged, sometimes intentionally, sometimes unintentionally. I think this is a very nice way to picturize our cellular niche. Thanks to one of our junior colleagues in the lab, Mani, who actually suggested this to me. I am Bhupesh, and today I have just 20 minutes to share our working model with you on a very important topic of this century probably, and that is LongCovid. To start with, I have some disclaimers. Most of these ideas that we are going to discuss here today are based on preliminary data generated with very small sample sizes, hence results should be considered carefully. We are here today to discuss our ideas on LongCovid and how research is shaping along these lines. Today's presentation is carefully designed for general non-scientific audience, not having much of in-depth knowledge in the field. Hence, those who have expertise on this field might find certain topics too superficial. Today we are going to discuss 4 major points: the term LongCovid, our ideas on development of LongCovid, and the comparison between LongCovid and ME/CFS, and where do we stand now. Today we are not just talking about fatigue or post-viral fatigue. We are going to talk about post-COVID or LongCovid, which in my opinion is much more than just fatigue. But I do not think that post-COVID and LongCovid are the same. For me, post-COVID is more like the effects of a severe COVID infection, which might be the case with patients with initial severe SARS-CoV-2 infection, those who were hospitalized. But LongCovid represents mostly the consequences of COVID-19 infection in patients having mostly an asymptomatic or very mild first infection. I'm not suggesting any names here. That is not my expertise. But maybe experts in the field should think about it. So, what can cause LongCovid? There are potentially 3 possibilities. The first one is severe SARS-CoV-2 infection and subsequent effects, including the widely discussed cytokine changes. This possibility is very widely discussed nowadays, but we have to consider the fact that majority of the patients infected with SARS-CoV-2 do not develop LongCovid. So this possibility is probably not the ideal one. There are 2 other possibilities which fits to our working model. The second possibility is that SARS-CoV-2 persistence in localized tissues and afterwards reactivation. The third possibility is that SARS-CoV-2 as an initial trigger, which afterwards reactivate persistent or latent viruses like herpes viruses, endogenous retroviruses, etc. So here is our working model. This is not a hypothesis, as it is based on our own ongoing work and is constantly evolving. I believe that there are 2 different forms of LongCovid or post-COVID. The first one probably starts with a very strong dose of virus infection. For example, in those who work in the healthcare system and frequently get exposed to infected patients. Here the virus dose is high and probably it rapidly takes over the host cell. These patients will mostly be hospitalized because of severe clinical conditions. There will be severe cytokine response and subsequent tissue and organ damage. There will be longer recovery time in these patients with various clinical conditions which are basically a result of localized tissue damage from the severe COVID-19 infection. The other type of patients will have very low infectious dose. Their host innate immunity probably is very strong, which keeps the infection at a very low level. Probably these patients have a predisposition to latent virus reactivation events. There will be either completely absence of SARS-CoV-2 infection. This means that the infection comes in but completely get removed, or there will be very weak SARS-CoV-2 infection. And there is a possibility of persistency also with low or no humoral immune response. There might be tissue-specific reactivation events of latent herpes viruses, which is again interdependent on each other. The weak virus infection can reactivate the virus, and the reactivated virus can help the SARS-CoV-2 to go into persistency. There will be weak or very specific cytokine response in these patients, and there will be tissue-specific damage causing immune dysfunction of the autonomic nervous system, which will have long-term effect causing autoimmune response, metabolic alterations that we see in chronic fatigue syndrome patients. So patients, the symptoms will be comparable to them. You must be wondering from where all these herpes virus or latent virus stories started. At the very beginning stages of the pandemic, roughly around March, April of 2020, many clinical case reports were published showing infection of different herpes viruses in patients with COVID-19. Clinicians were not aware of potential virus reactivation at that time, so they described all of these as virus infection or co-infection. We were a bit surprised to see such an event. We realized that it might be a cause of reactivation than a fresh infection, particularly in days of social distancing. This reminded me a very unique and unexpected scientific story of my life. So the story started when I joined the labs of Professor Thomas Ruttle in 2009, who at that time was studying a bacterium called chlamydia for its role in cell death. This bacterium strongly inhibits cell death. So Thomas asked me if I can infect these cells with a virus which actually induces the cell death, and then if I can see if still the chlamydia can prevent the cell death. This would be a natural stressor for chlamydia. By the way, chlamydia is a gram-negative obligate intracellular bacterium. Chlamydia infection is one of the most frequent bacterial infection in the human being. Chlamydia trachomatis infection is actually one of the most frequent sexually transmitted disease also. It has a biphasic life cycle in the form of elementary bodies and reticulate bodies. In the structured illumination microscopy over here on the right hand side, you can see these elementary bodies in pink color in a cell culture, in a human cell in culture. I had experiences with HHV6 before, and I knew that it induces cell death. So we took some cells, infected them with chlamydia, and then threw some HHV6 viral particles into the culture. Next day, we were all thrilled to see the effects, but we were super disappointed as there were hardly any chlamydia growing in it. Rather, there were huge, persistent, non-infectious inclusion bodies were there in these cells. You might be thinking that HHV6 stopped bacterial growth, so it's all good. But not exactly. These persistent bacteria are not dead completely, and they cannot be treated with antibiotics. At any moment of time, when the physiological conditions allow them, they can come back or revert back to the normal infectious form of the chlamydia. Now, on the other hand, when we took cells carrying latent HHV6 genome, for example, the virus that is integrated within our chromosome, and infected these cells with chlamydia, we saw suppressed chlamydial growth but reactivation of the dormant virus. Now we know that this happens when one of the bacterial protein or one of the chlamydial protein changes the structure of our telomere. It allows the latent virus from our telomere to come out and starts growing. Now, if you are thinking that it is way too complex and how two pathogens can really cross-talk with each other, you are not alone. We were criticized badly 10 years back by the reviewers. But then now, after almost 10 years of work, we can explain you many of these phenomena. So it all revolves around mitochondria. Chlamydia is basically an ATP parasite. Chlamydia infection protects the host mitochondrial architecture by allowing mitochondrial fusion. Here in these cells, you can see a different mitochondrial morphology in cells having chlamydial infection. You can see those pink elementary bodies in these infected cells with very long networking mitochondria inside. Chlamydia needs a lot of metabolite and ATP from the cell, so it needs mitochondria to do that job. But on the other hand, HHV6 is a slow-growing virus. It needs to protect itself from the host cell innate immune response to survive inside the cell. And hence, it doesn't like fused and good mitochondria. So it has its own machinery to fragment the mitochondria. If virus wins and the mitochondria is fragmented, chlamydia cannot grow, so it hides. When bacteria wins or grows inside the cell, unintentionally, it allows the dormant virus to become active. Most people think that if a latent virus reactivates, it produces new virus particles and infects new cells and spreads throughout the body. That's absolutely not true. When a latent HHV6 reactivates, there are in fact many possibilities. The first possibility is that the host innate immune response wins and the reactivated virus is not allowed to grow anymore. There is no transcription, no expression of viral proteins, and everything subsides. The second possibility, as I said, there is full-blown viral reactivation. There are formation of viral particles, which infects new cells, and the infection goes on. The other possibility is a leaky infection where there will be production of some isolated viral gene expression, which in fact can form antigen and allow the host immune response to develop. Sometimes it can also allow autoimmunity to develop by molecular mimicry. It can help in immune evasion. It can also transactivate some of the cellular genes. Sometimes the reactivation of the virus can cause chromosomal instability also, and as the HHV6 particularly is in the telomere of the chromosome, it can actually cause premature chromosomal senescence also. It can also, if the virus comes out of the chromosome, it can also cause telomeric truncation also. And in extreme case, it can also activate proto-oncogenes and can contribute to cancer development. So herpes viruses are some of the most underestimated viruses because they are very common, and as they are common, we think that they are harmless. But the fact is different. These dormant or latent herpes viruses often reactivate under various physiological conditions, and they affect host cell in various ways. Now, if we look into the prevalence of these herpes viruses in general population, we can see that, for example, the herpes virus type 1, HSV1, is almost 66.6% in general population. HSV2 is a little bit lower, around 13.2%. The varicella zoster virus was very high before 1995, but after the vaccination started, its prevalence has gone down. The cytomegalovirus prevalence is up to 50%. EBV, which is a tumorigenic virus, herpes virus, is there in up to 90% of the population. The Kaposi sarcoma virus is also ranging from 10 to 90% in different geographical regions. The HHV6A, 6B, and 7 highlighted here in yellow are very unique viruses because they can also be inherited. You can get the virus genetically inherited from your parents also, and prevalence of these viruses are also extremely high, from 85 to 90% in the general population. So we think that these herpes viruses, which are so prevalent in general population, must be contributing from time to time to our own cellular physiology. So we came up with a pilot experiment based on our initial hypothesis that it might be possible that the SARS-CoV-2 is somehow reactivating these latent herpes viruses. And this is what we also wanted to validate for the study of MECFS. Then during this time, people started realizing delayed appearance of MECFS like symptoms in post-COVID patients, long-COVID patients. So we went ahead and infected cells carrying latent herpes viruses, particularly HHV6, because this is our most favorite virus, let's say. And then we wanted to see if the SARS-CoV-2 infection is doing something to the HHV6 reactivation. So what you see here in your screen are U2A cells. These are bone osteosarcoma cells, and these cells reactivate HHV6 quite nicely in culture, so we use these cells for our studies. So these cells are genetically altered to carry a reporter latent herpes virus 6 viral genome, which actually produces the green protein, the GFP protein, only when the virus is reactivated. So here are two representative images of these cells infected with SARS-CoV-2. These cells have very low ACE2 receptors, so they do not get infected heavily until we put the ACE2 protein in the cells. That's good for us because that allows a buffering time for the interaction between the virus and the host cell, like it normally happens within our body. Here are the virus spike proteins, so that you can see that the cells that are infected with the SARS-CoV-2. You can compare the virus growth to this beautiful image from Goldsmith et al. Paper published in Lancet in 2020. SARS-CoV-2 grow within membrane-bound vesicles, like I showed you for chlamydia. You can compare with the bacterial structure. Now you can see that there is a reactivation of HHV6 in some of these cells, which are green in color. The interesting fact here is that the cells which are reactivated mostly do not carry SARS-CoV-2 infection. You cannot see spike proteins inside these cells. It seems that some of them do have small vesicles, like the infection started in these cells, but without much of virus spike proteins. We are working at single cell level to understand why virus reactivating cells do not allow SARS-CoV-2 to grow. Is it persistency that we are predicting or we are talking about, which actually inhibits the virus growth inside the cell? Future will answer those questions, but for the time being, it seems that there is a crosstalk between SARS-CoV-2 and HHV6. The SARS-CoV-2 infection reactivates HHV6, which is possibly helps in suppressing even the SARS-CoV-2 infection. We do not believe that latent herpes virus reactivation is widespread and it can be detected everywhere, including blood and serum. This is not the case. For example, in MECFS patients, we found very strong HHV6 reactivation in cranial nerve ganglia. In these images, you can see the viral microRNA being expressed in these nerves, which potentially explains the autonomic nervous system dysfunction in these patients, explaining the clinical effects on heart, lungs, kidney, digestive system, or even construction of blood vessels, etc. So now the big question that we are discussing today is whether long COVID patients are similar to MECFS patients or not. Frankly speaking, we have preliminary data that suggests that the first stages of long COVID is not exactly comparable to MECFS. But possibly, if the symptoms and clinical conditions persist for a long time, the long COVID patients might develop into MECFS. So what is common between long COVID and ME/CFS? We are here able to show that herpes virus reactivation is a common thread or signature between ME/CFS and long COVID. We are able to see that many of the ME/CFS patients and the small number of long COVID patients that we have managed to test so far have antibodies against many of the herpes virus DUTPages. DUTPase proteins are highly conserved among herpes viruses and are expressed during early stages of virus reactivation. These are the proteins which are necessary to prevent the DUTP to be incorporated into DNA molecules. Normally UTP should be there in the RNA molecules, but when the DUTP is incorporated into the DNA molecules, it can induce innate immune response. So these proteins are there, very conserved during the process of evolution, and which actually prevents the DUTP to enter into the DNA molecule. Now, healthy controls also have these antibodies, but not against multiple herpes viruses, particularly not against the EBV DUTPages. Here is the heat map that you can see where the pattern in the first 25 ME/CFS patients from the top is comparable to the 5 long COVID patients that we have tested so far at the bottom. But these patterns are not comparable to the 20 healthy controls which are there in the middle, because of the presence of antibodies against multiple different herpes viruses, including EBV, HHV6, HSV1, 2, etc. We believe that the presence of antibodies against these proteins also mean that not only the antibody is there are these herpes virus proteins also somewhere in the body, or maybe also in the serum. We can also show that these individual herpes virus proteins or DUTPages are alone, they are not very effective in inducing mitochondrial fragmentation, but together with other herpes virus DUTPages, that means when multiple different herpes virus DUTPages combine, like EBV with HHV6 DUTPase or EBV, HHV6 and HSV1 DUTPase in a single cell, they are very effective in altering mitochondrial architecture and possibly its metabolism of the host cell, and also can induce host cell senescence in a long run. So where are the differences then? Recently, we looked into 240 different antibodies in human serum using a very high throughput approach. The serum antibody profiles of ME/CFS patients suggest high degree of overlap between inflammatory rheumatoid diseases, autoimmune diseases like SLE, Hashimoto thyroiditis, celiac disease, neurodegenerative diseases like multiple sclerosis, and ME/CFS. We found some of the antibodies against some of the components like antibody IgG against the single-strand DNA is very high in ME/CFS patients, statistically significant enough, which is not the case in long COVID patients. For example, the IgM response against the myelin basic protein is very high in ME/CFS patients in comparison to the healthy controls, which is not the case in the long COVID patients. So tests in long COVID patients doesn't point towards the same as we see in ME/CFS patients. So autoimmunity might be a consequence of pathological changes in ME/CFS, which is too early to see in long COVID patients. To test our working model about long COVID that I just explained, we are undertaking several projects. For example, we are looking into the proteomics of the serum that we find in ME/CFS patients and long COVID patients, and how these proteomics differ or overlap between these two conditions. We are looking into a single cell at a RNA dynamics label called SLAMSEq, using a technique called SLAMSEq, to understand individual blood cell transcriptomics in long COVID and ME/CFS patients. We are also searching for novel antibodies which may not be studied, or we cannot study in using large-scale high-throughput approaches. So if we can find such antibodies against infectious molecules or non-infectious molecules in the serum of long COVID or ME/CFS patients. We are also looking into a very core component of innate immunity called RNase, called RNase L, whether it has some role to play in innate immune dysfunction in long COVID and ME/CFS patients. So all these projects are funded, or some of them are even not funded. So we are constantly looking for financial support. It would be exciting if we can also look into various other aspects of these two conditions, long COVID and ME/CFS. But there is always financial constraints which keeps us focused on some of the very core aspects of our work. So I would like to thank our collaborative partners and funders within Germany and outside Germany. Without their help, this project or our work is absolutely impossible. We are also looking forward for new clinical partners in other European and non-European countries so that we can expand our work to patient cohorts outside of Germany. And we are supported by some unusual small, large foundations, and also the ME/CFS patient community, who constantly provides some financial support to keep us going in our quest to solve this disease. So thank you very much for your attention, and I would really love to take questions and discuss the subject maybe in more detail with you. So I'm looking forward to it.